Well, welcome to the last chapter of the semester. Wow, did you think you'd make it this far? I have to congratulate you. Awesome job so far. Let's keep it going. One more chapter. Don't let up now. The end is in sight. Keep it together. So, this is all about the sensory systems of the body, and we need to redefine some terms that we have discussed before, so hopefully they'll ring some bells. We have sensory receptor, which is a structure that is specifically designed to detect a particular stimulus, and this can be a bare nerve ending, or it could be a specialized cell that is used to detect a particular stimulus. And then very nearby to that cell will be a neuron and its dendrites. So, what do receptors do? Well, they change the very nature of the stimulus. So we detect things, we see things as green, as yellow, as tasting a certain way, hearing certain information, but after we detect it, we convert it into action potential. So this idea is called transduction, to basically translate or convert one type of signal into another. All right, so think about your air conditioner. Your air conditioner has a coil, at least the old ones did. And this coil would wrap around in a circle, and the middle part would have a contact, and then there'd be a contact sitting out some distance away. And so as things got progressively hotter, the coil would expand, eventually making contact with the contact that's way out here. And when it did, it completed the circuit, turns on your air conditioner, things cool down, the metal contracts then, breaks the contact, and you stop the air conditioning. So that's a very simple way of transducing heat into electrical energy. And this is the same type of situation. We are going to develop a potential on the receptor in order to initiate an action potential. And so we might do that with bare nerve endings, but with specialized sensory cells, we will probably see the result would be a release of neurotransmitter, which will then act on the subsequent dendrites or nerve cell body found after that specialized sensory cell. So sensation is just an awareness of a stimulus, and it can be filtered out in the brainstem, quite possibly. pH, body temperature, we constantly have our homeostatic processes that deal with changes in these. But what we're comparing sensation to is perception. And perception is more than an awareness. It's an understanding of what's causing the actual stimulus. And so then we'll move away, we'll do something about it, perhaps a long-term fix. All right, so sensory receptors transmit 4 types of information. Why would this be needed by the brain? All action potentials are exactly the same. All we can do to tell how they're different is based on these 4 types of information. Modality, and we'll define these further here in a second. Where the information is coming from, how strong it is. This goes back to that refractory period that we discussed in chapter 12, or its intensity, its frequency, and then how long it lasts. All right. And so when we talk about modality, we mean what is the stimulus? What type is it? So the brain is already coded to respond to specific stimuli in preference to other stimuli. Okay, so things like vision, hearing, and taste are going to be responded to fairly quickly. And remember, all action potentials are identical. Can't help but get away with that. Where is this at? Not what type of signal is it, but where is it coming from? And so the brain is going to work to identify where it's coming from, and if it's in an area that is particularly useful to the brain and its maintenance of safe environment, then it's going to respond to that much more quickly. So part of this has to do with what's called the receptive field. Remember, an axon is leaving the neuron and then can branch into many branches. And where those branches end up at and the area that they are responsible for is called the sensory field. So in the fingertips, you're going to have incredibly small sensory fields or receptor fields. In the stomach or on the back, you're going to have huge sensory fields. And this has to go back to that thing we talked about in chapter 13 called 2-point discrimination, where you take a compass and you put it, the 2 points of it together, and then you start slowly separating those 2 points. And when you can finally identify it as 2 separate stimuli, that's the width of your receptor fields. On the fingertips, they'd be incredibly small. On the arm, they'll be much larger. And here is the notion here of the 2 receptor tips. If you have a large receptor field from a neuron, then you would not be able to detect that those are 2 separate tips. Versus in your fingertips, you would probably very quickly be able to tell that there are 2 separate tips that are piercing your skin. The intensity of the signal is which fibers are sending the signal, how many fibers are doing so, and how fast are they firing. This is what we were talking about with the refractory field. So once again, the brain's going to pay attention to those that are of a higher frequency and those that encompass more fibers firing. And finally, how long does this stimulus last? Does it retain this high frequency of stimulation over a longer period of time? We're going to pay more attention to those, whereas if the frequency kind of dwindles off after a bit, then we won't pay as strong attention to that particular stimulus. Okay, and it's just a general law of neuron firing, of action potential firing, that as the action potentials are prolonged, the stimulus is prolonged, the neuron will fire in a slower and slower pattern and will become less and less aware of this stimulus. All right, so we can classify receptors by modality: thermo, photo, noso, chemo, and mechanoreceptors. We'll talk about those. By where they are, where the origin is for the stimulus, and we've discussed these previously, chapter 13, 12 or 13. Okay. Or by distribution. Is it a general sense, which are very widely distributed, or is it a special sense, meaning hearing, seeing, or smelling, or tasting? So the general senses are the idea that the receptors are very simple. They're called mechanoreceptors, and they respond to mechanical stress, like the stretch of a muscle, the stretch of skeletal muscle in the joints would be of particular interest to the cerebellum, right? So the brain's going to make sure the cerebellum is aware of that. There are a few sensory fibers, right? And in these types of endings, these types of mechanical receptors commonly found in joints and the muscles and lining every square inch of your skin, we will see there are 2 types. There are unencapsulated nerve endings and encapsulated nerve endings. I don't want you to get too wrapped up, no pun intended, in these particular types, but you should understand that dendrites that are not wrapped in connective tissue are unencapsulated, and this would include free nerve endings for pain and temperature. So we see these just raw nerve endings that are projecting right up underneath the epidermal layer of the skin. The tactile disc that we've discussed before when we discussed the 5 layers of the epidermis, the tactile disc are right near, or right in the innermost layer, right, in that stratum basal. And hair receptors that are functioning to monitor the movement of the hair. So that's these top 3 that are unencapsulated. The rest of these types of receptors are termed encapsulated receptors, and they're wrapped by some type of glial cell perhaps, or even some connective tissue. And this helps to enhance their sensitivity to the stimulus. Some of these may be responsive to deep pressure. So it's going to help with that response of the deep pressure. It's going to be capsulated and covered so that the deep pressure will have influence on that connective tissue that surrounds it and cause it to deform, and that will stimulate the neuron that's inside it. So we have these Meissner corpuscles. They're for light touch, just like the tactile disc are. The Krauss-in bulbs are also tactile, but they'll respond to a little heavier pressure and touch. The Pacinian corpuscles are due to deep pressure, and the bulbous corpuscles are due to some very heavy touch. Okay, so how are these projecting? Just a reminder again of how these work. The first order neuron carries the information from these receptors to the spinal cord. And they're going to be very fast typically. However, temperature and some pain sensations are transmitted via the type C or unmyelinated fibers. Then the second order neuron is going to carry that information from the entry into the spinal cord up to the thalamus. And then we will have the third order neuron that will carry that information on up to that particular region in the precentral gyrus, so the somatosensory cortex, postcentral gyrus, or the somatosensory. Okay, so I just realized that I left off a little bit about the modality of receptors that I wanted to talk about more. There are 5 types or 5 classifications of sensory receptors in the human body. The first type we're going to talk about are thermoreceptors. They're found all over your skin, and they're there to detect changes in temperature. Then we have the photoreceptors that are found, of course, in your eyeballs, that are there to detect light, changes in light patterns. And then nociceptors are the names of the receptors that detect pain, which isn't really a sensation. It's tissue damage that you should put if I ask this in a short answer question. So nociceptors detect tissue damage, which relates to pain. And we've got a whole video coming up on pain here next. Chemoreceptors detect chemicals or ions, and so these are valuable in the sensations of smell and taste. And then the mechanoreceptors that we talked about thoroughly that respond to mechanical stressing and are found in joints and also along the surface of the skin.